Okay, so what happens now when there's injury? So the first thing that you see is vasoconstriction. So in vascular constriction, this will result from muscles contracting. Now, why do the muscles contract? So the muscles contract because of two things. The first thing is that the early response to injury will mean that you have vasoconstrictors from nerves. Okay, so from nerve endings that are close by to the injured area. So this is known as the neurogenic response. So once you have those vasoconstrictors, you also have them causing muscles to contract, and this will lead to the constriction of the vessel. And the muscle contraction itself is a myogenic response. Not only do we have this, but we also have endothelin. So endothelin is also produced by the endothelium as a vasoconstrictor, causing even further vasoconstriction. Secondly, you have now the formation of the platelet plugs, the primary platelet plugs. So in this case, you're going to have adhesion and aggregation. In adhesion, the first thing you're going to see is that the platelets will now be exposed, okay? They will be exposed to the collagen, okay? And what you will see is that the injured cells will produce substances called von Willebrand. So the von Willebrand factor is then going to bind to the collagen, and then as we earlier saw, we noted that the platelets do have receptors for von Willebrand, so they will also bind. This is von Willebrand, so they'll also bind to the platelets. So this binding that you see of the von Willebrand to the platelets, as well as the external component here, you will note that it will cause the surface to sort of be sticky, so to speak, if I can pick a word. It makes it sticky because it can bind to the external component as well as the platelets. So you can call this adhering or adhesion. So adhesion is therefore just the sticking of platelets to non-platelet surfaces. Okay? The next thing that you're going to see is aggregation. So aggregation has got the activation stage. In the activation stage, you will observe that when the platelets come into contact with the damaged vascular surface, they will undergo a conformation, which is a change in structure. And this change in structure is such that they will swell up, and then they will irradiate pseudopodia, all right, forced legs, yeah? All right, so once they turn into this, or they conform into this, they would then release granules, okay? And then this release will cause them to become sticky. They will become sticky with each other, all right? So the stimulation of the receptors on the platelets by von Willebrand will also signal activation of phospholysis enzymes. Phospholipase enzymes is going to activate the arachidonic acid, and once this is activated, it's going to also activate thromboxane A2. Now, this is a vasoconstrictor, but it is also a platelet aggregator. Okay, so it reacts on nearby platelets, activating them and causing them to attach to each other. Now, when platelets attach to each other, we call this aggregation. Okay, so first of all, we have them conform, they release granules, become sticky. Also, we have the activation of phospholipase because of the von Willebrand activity, and this activates thromboxane A2, which is a vasoconstrictor but also a platelet aggregator, causing aggregation of platelets. So what you'll be seeing really is more platelets coming to this area. Just going to make our vessel larger. Okay? So once that happens, we can also identify the fact that the von Willebrand, upon activating receptors, in particular the GPIB receptors of the platelets, further granules are released. Okay, so first of all, we talked about activation of phospholipase, but it also causes granule release. So these granules that are released include those ones that we're talking about, which are the dense and the alpha. So the calcium from the dense granules are able to bind coagulation factors to the surface of the platelet. Okay? Let me say that again. The calcium that is from the dense granules will bind coagulation factors to the surface of the platelet membrane. Then we also have other things happening, such as serotonin, which is a vasoconstrictor, so more vasoconstriction is going to happen. And ATP is also a platelet activator. So all of these will be released to enhance what is happening here, the aggregation of platelets. So the aggregation will allow for this very loose plug to be formed. And this plug is what is known as the primary platelet plug. All right. So that was number 2. So number 2 was formation of the platelet plug, which involves adhesion and aggregation. Adhesion meaning the platelets sticking to a non-platelet surface. Aggregation meaning the platelets sticking to each other. The third one is now what is known as coagulation. So what are we talking about when we talk about coagulation? So the surface of the platelet expresses proteins that favor coagulation. And the primary platelet plug, we see that the first thing that's going to form are fibrin strands through a cascade of reactions. And these fibrin strands, they're the ones that now produce a tight secondary hemostatic plug. So we're going to go through the stages that form this secondary platelet plug. So there are two sort of ways in which this can happen. First of all, there could be trauma to blood and trauma to the tissues. These are the two pathways. So these involve the intrinsic and extrinsic pathways that both have several phases. So in the next video, we'll be talking about the intrinsic and extrinsic pathways involved in making the secondary platelet plug.